Now that we've finished looking at the role of the liver, the gallbladder, and bile in assisting with the digestion of fat, it's time to look at the ultimate conclusion of digestion and of nutrient absorption. So remember that although the small intestine doesn't produce most of its own enzymes, it's actually the pancreas that produces the major enzymes that are responsible for the large-scale breakdown of pretty much every undigested nutrient that makes it into the duodenum. Now, by the time the pancreatic enzymes have done their work, most of the nutrients will be reduced to small fragments like this. So here we can see that this here is an individual nucleotide, which in this case it came from DNA, and again, DNA is made up of many of these. We can see here that this is a dipeptide because it's two individual amino acids, which falls under the category of oligopeptides, meaning short chains of amino acids joined together. And here we have two individual carbohydrate monomers. This is what we call a disaccharide or a pair of sugar molecules here. Now the problem is that even though these nutrients are significantly smaller than the originally full-size nutrients, they are still too large to be transported into the capillaries of the villi individually, and so your digestive system, or rather your small intestine, actually needs auxiliary enzymes to break these down further, and these auxiliary enzymes can actually be produced by the small intestine itself via special glands that are found in between the different layers of the intestinal wall, which is why we call them the intestinal interstitial glands. Interstitial is just a fancy word that means in between the different layers of the walls of your intestines. We can see that there are small enzyme producing glands embedded within the muscular layer of the small intestine. Now, for example, there are three that can address these small but still too large molecules and break them down into individual components. The enzyme that targets this particular pair of carbohydrate monomers, this is a molecule that we call maltose, which is actually made of two glucose molecules, and not surprisingly, the digestive enzyme that breaks this bond and separates them into two individual carbohydrate monomers is what we call maltase. Maltase digests maltose. Now, for short fragments of amino acids or short chains of amino acids, we have an enzyme related to proteases, which are what we call peptidases. So peptidase breaks down short chains of amino acids. The word peptide in reference to proteins, because another name for amino acid is monopeptide. And finally, this nucleotide can be broken down into three components: the nitrogenous base, the phosphate, and the sugar, which is broken down by the rather awkwardly named nucleosidase. So, once each of these auxiliary interstitial enzymes has broken down the nutrients that are left over from the major pancreatic enzymes that do most of the heavy lifting, now your nutrients are small enough and sufficiently ready for absorption into the capillaries that are found within the small intestine's microvilli. There's just one problem. This is the ideal process for sugars, for amino acids, and for nucleic acids, because every single one of these nutrients is able to dissolve in water fairly easily. But obviously you don't want to be doing this with fats, because blood is mostly water, and as we've just talked about, fats are almost entirely hydrophobic molecules, meaning that they will dissolve poorly or not at all when they are put into water, which means that your digestive system is going to need an entirely different transportation system for the hydrophobic nutrients that make it through your digestive system. And this transportation system are what we call your lacteals. So this is the structure of a villus, and we can see that there are capillaries in here, but we can see right in the center that there is a second tube, and these are what we call your lacteal system. And the general idea is that lacteals exist as an alternate transportation system for any material that is hydrophobic, such as fats. But of course, fats are not the only substance that dissolves poorly in water. The other major example is cholesterol, which of course is the steroid that is found in egg white, and certain non-polar vitamins also dissolve poorly in water, such as vitamin A and vitamin K. And in the same way that the hydrophilic nutrients that are able to dissolve very well in water, will diffuse from the inside of the small intestine into the blood vessels and then be actively transported if the concentration is not favorable. The same thing occurs with hydrophobic nutrients that don't dissolve well in water. Only their transportation would bypass the blood vessels entirely and enter the lacteal system, where they become part of a mostly hydrophobic fluid that doesn't mix well with water, which is what we call lymph. So lymph, if you have actually gotten a cut and have seen a fluid that is not blood, but is actually kind of a milky white or sometimes off yellow color leaking out, what you are seeing is actually lymph, because not only have you cut blood vessels, you also have cut lymphatic vessels. Now, just like the blood vessels, or rather like all blood, the first destination of lacteals, once they leave the digestive system, is going to be to the liver. So we can see, if we look at the flow of lacteals out of the small intestine, we can see that the first major arrival location is going to be the liver. And this is where a lot of nutrients, especially fats and cholesterol, are going to be stored for future use. And for fats that are not healthy at all, such as trans fats, this is where detoxification is going to occur. And again, during the process of detoxification, either the harmful substance is either outright broken down into substances that are less harmful, or they are modified in a way that makes them more easily able to dissolve in water, and therefore more easy to remove via your urine. Now, we can see that lacteals actually are a very small part of a body-wide transportation system, and if you remember that the name of the fluid that moves through lacteals is called lymph, not surprisingly, the system that transports lymph, not just out of the digestive system, but all throughout the body, is what we call your lymphatic system. And the lymphatic system is actually not primarily designed to transport non-polar substances. This is actually a subsidiary benefit of it. The lymphatic system actually exists to primarily transport your body's white blood cells, because for the most part, white blood cells are significantly larger than red blood cells, which makes them unable to use the capillaries because they're simply too big for them. And technically that means that the lymphatic system is actually part of the immune system. However, just because the primary function of the lymphatic system is to transport your white blood cells does not mean that it can also have this subsidiary benefit of transporting non-polar nutrients. In fact, this is quite essential, because any of these hydrophobic nutrients is not going to be able to dissolve in water, which means if a large amount of fats, cholesterol, or non-water soluble vitamins build up in your blood vessels, you can actually get a blood clot, and this could potentially kill you if it happens within your heart or your brain, and therefore the lymphatic system is absolutely essential as a parallel transport system for hydrophobic nutrients. This concludes our section on the roles and functions of the small intestine. Before you move on to our series on the large intestine, you can take a look at this case study here that looks at a virus that specifically infects the digestive system, and you can investigate what the causes and problems of such a virus might be before taking a look at the practice problems below, testing your knowledge of the small intestine before you can move on to the series covering the large intestine.